Hi, good evening. My name is Gabriela Sanchez. Today is Monday, December 7th, 2020. I'm going to be doing the motility test lab experiment. I've gathered all of my materials. I have both of my both of my plates. I have a candle that's already been lit. I have the motility test agar vials. I have two of them. One, they've already been labeled. One is for E. coli and the other one is for S. epidermidis. I have two paper clips soaking in alcohol, and I also have bleach to place them after I'm done with this part of the experiment. So I've already washed my hands. I've disinfected the area. I straightened two paper clips. I don't have the large ones, so I have the small ones, but I'm going to be using the clamps to help with the stabbing of the motility test tube. So the first thing I'm going to do is I'm going to get the E. coli test tube, and I'm going to open it. I'm going to pass it through the flame to sterilize it before I introduce anything to the vial, or to the test tube rather. Okay, I've already passed it through the flame to sterilize it. I'm going to remove a paper clip, and I'm going to use the tweezers because I don't have the large one. So I'm using this to kind of give me a little bit more length. I'm going to uncover or remove the lid from the E. coli plate. And I'm going to collect colonies with the tip of the paper clip. Okay. Not sure if I'm supposed to go through it or not, but I'm trying to gather as much from here. And what I'm going to do, I'm going to recap that. And with the tweezers, I'm going to grab the tip and stab the test tube. It's not really formed. It's a little liquefied. Okay. So it's not really hard to stab it. It's kind of easier to put. I'm going to put this in the bleach. To soak there. And before I recap this, I'm going to pass it through the flame once more. I need to light my candle again. I do apologize. It turned off. All right, turning that back on. And before I light this, or before I recap it, I need to pass it through the flame to sterilize it and recap. I'm going to do the same with the S. epidermidis test tube. I'm going to remove the cap, pass it through the flame to sterilize, and uncover the plate. And I'm going to grab my other paper clip, make sure that I don't have any excess liquid or alcohol dripping. And I'm going to collect some colonies from my plate. And with the help of my tweezers, I'm going to again stab my test tube, the motility test tube. Trying to get as far down as I can. And I'm going to soak this in bleach. And before I recap it, again, pass it through the flame to sterilize it and recap. I'm going to put those my test tubes to incubate for an additional 48 hours, and then we'll see the results video to follow. I'm gonna take pictures of it, and I'm gonna wipe down my work area with the bleach solution, wash my hands, and return whatever I need to reuse for future experiments. That concludes my video.